Good morning, and thank you for inviting me to speak at the Healthy Skin Microbiome Summit. On behalf of the National Biofilms Innovation Center, NBIC, I would like to talk about new product development considerations to foster healthy skin microbiome and about partnering with academia for successful collaboration. I am Katerina Steventon, a senior innovation consultant for NBIC in the UK. So first question, what is a biofilm? If microbiome is the total genetic composition of all species which are present on the skin, then biofilm is the predominant way in which the microbiome establishes itself in any ecological niche, as well on the skin. Biofilm is a well-characterized mode of growth of both single and multi-species populations. It is associated with community and cooperative behaviors, which are very different to individual bacteria living as single cells. Bacteria become aggregated together and protected by a self-produced slimy extracellular matrix, so-called EPS, which is comprised of various biopolymers. Biofilms are more tolerant to antimicrobial compounds. And you can see on my slide the biofilm life cycle, which starts with reversible attachment of the microbes to the surface, which leads into irreversible attachment, the biofilm growth, biofilm maturation and dispersal. And biofilms are relevant across multiple industry sectors, and those sectors closer to skin health would be the gut health, where biofilms are positive, and wound care, wound healing, where biofilms are negative. So NBIC, who are we? We are an innovation knowledge center and a hub for accessing expertise and capability, early scientific knowledge and insight to generate IP, and understanding innovation capacity in the UK. We catalyze the growth of expertise in both science and technology and deliver world-class science as well as scientists focused on breakthrough innovations and deliver also value both to the economy and the society. NBIC is built on three key pillars, which are research, innovation, and training. In terms of research, we have postdocs in 4 core universities who work on fundamental biofilm research. We also have research strategy in place, which has been endorsed by International Advisory Board. And we have built a common ontology language with our UK researchers. In terms of innovation, we have a portfolio of 83 proof of concept projects which are funded. We have carried on with 4 in-person workshops resulting in policy papers and roadmaps pre-pandemic. We have also visited 250 companies and research institutions, and our innovation is truly led by innovation team and expertise. Our industry engagement strategy is endorsed by Industry Advisory Board. In terms of training, we've built a community of postdocs. Our BITE, Biofilms Innovation, Technology and Engineering, is a community of PhD students. And we have performed skills gap analysis with our industry partners. How do we facilitate innovation? Well, to start with, by building an environment and creating the right tools. We explore unmet industry needs by talking to the industry through workshops, dialogue, networks, and roadmaps. We also carry out funding calls and help collaborations and support connections to advance our research. We exploit emerging breakthroughs, drive the research agenda, and influence key stakeholders, be that regulators, funders, or society at large. Our academic partners. So we are building a national and international community of biofilm researchers and have had 5 accession rounds. NBIC is a club of 63 UK research institutions at present. We also have a memorandum of understanding and links with leading international centers. That is CBE, the Center for Biofilm Engineering in Montana in the US, and the Singapore National Biofilm Center, CELSI. We are a club of more than 250 industry partners and growing. So a growing network of companies and organizations with needs and opportunities. So in your remit, to name just a few, for example, BriCure is an SME that develops vaginal microbiome related products. Few ingredient suppliers there, for example, Croda and Simrise. Finished product manufacturers, Boots, Unilever, GSK, and testing houses like LabSkin or Bluetest or Perfectus Biomed. We work overall in strategic interventional themes. Prevent, detect, manage, and engineer. Prevent doesn't apply too much to skin health. It is a knowledge-based design of surfaces and materials. Anti-adhesion surfaces that exploit surface topography or smart delivery of antimicrobials via coating and surface functionalization. Detect, which is applicable to skin health, is about accurate, quantitative, actionable detection or sensing of biofilms. So rapid and real-time and in situ biofilm detection is a key unmet need. Manage is also applicable to skin health. It's about killing, removing, or controlling established biofilms, exploit their life cycle dynamics. So it's targeted disruption of biofilm signaling and mechanisms of development, and also enhanced translation of anti-biofilm strategies through improved lab models. Engineering is about controlling and direct complex microbial community processes in process applications. So bespoke biofilm model systems are needed for understanding complex microbial communities and big data solutions to provide improved engineering platforms. There are also bigger cross-cutting research themes which apply to us. So microbe interactions, microbe metal material interactions, antimicrobial resistance, mathematical modeling and big data, and model systems, be that lab to real world or standards and regulations. So partnering is one of our major initiatives. It's within the academic and industry partners of our network. And our partnering searches start either with the unmet industry need or with the technology or capability opportunity. It's usually 50-50 academic and industry partnering search. In the recent time, we've engaged 30 academics with over 200 tangible follow-ups. So when the partnering search starts with unmet industry needs, we communicate this to the academic network and connect to the solutions or offers. If it starts with technology or capability, we will develop a broad summary and then connect with industry partners. This could be for collaboration, proof of concept project application, or just a dialogue. And all of our innovation team are industry specialists. Our proof of concept portfolio. So over the 4 proof of concept calls, we have awarded 83 projects in various sectors. And if you look at the middle of the graph, you see that health is very popular. Hygiene to the right, and again, in your remit, oral care and personal care, we have just grown these sectors. In terms of POC application themes, prevent and manage are very strong. Engineering and detect, we had to work to increase these. But I would like to give you some examples of proof of concept for awards. University of Bradford and NSME Verostatic Shield have been awarded POC. It is the development of an electrospan antimicrobial coated tampon for management of bacterial vaginosis. University of Edinburgh, Bear Valley Ventures and Aqualution have a POC on development of an in vitro human skin biofilm model for testing active ingredients for hand hygiene. And the last example is University of Glasgow and Unilever with Penrose Bio, working on incorporating a quorum sensing blocker, lactams, into topical treatment to control mixed biofilms on keratinaceous infections. So pre-pandemic as well as now, we have carried through workshops and webinars to discover unmet industry needs and further the collaborations between academia and industry. We have held 3 workshops so far: detect, then engineer, manage, and we are yet to have the prevent workshop later this year. We curate the tables and aim for attendee balance, 50-50, or at least 40% from the industry across all sectors and 60% from research institutions. And in terms of our webinars, I would like to bring your attention to the webinar held with the CC UK, the Cosmetics Cluster UK, on the science behind skin microbiome and biofilms, which we have run in December 2020. And also the recent Innovation in Deodorants webinar run in April 2021. They are all these recordings are on our website. Just going back slightly on the biofilm detection workshop and the workshop outcomes. Again, the report is available on our website. And the key needs which came out of these workshops. Truly, everybody is searching for rapid in situ, point of use, new and emerging technology to detect biofilms. We also want to understand the biomarkers that are definitive for biofilms, be that in skin, blood, industrial fluids, and other secondary media. It would be great to finally detect and characterize when a biofilm transition happens from a healthy to an unhealthy and pathogenic state. So the state of dysbiosis, for example, in skin disease or in wound healing. And it would be great as well to detect and confirm the presence of a biofilm in a standardized and reproducible manner, which is also acceptable to regulatory agencies to make a claim. Few notes from the biofilm management workshop. Again, the industry and academics are searching for improved models and methods for characterization, visualization, and detection of biofilms, which are relevant to the real-world context. They are standardized and not too expensive, accessible. They also praised cross-disciplinary collaboration, which NBIC offers, and still calling for improved collaboration across the industries. So be that industry, academia, industry to industry cross sectors, or bringing in the regulators. They are asking for clarification of pathways from industry regulators, and NBIC has a leading role to play in this. In terms of our recent webinars and blogs, I will just mention one which is relevant to this industry. It's innovation in deodorants. So starting with the consumer, we understand their concerns, feeling self-conscious and isolated with malodor. And we understand that the axillary microenvironment is a hotspot for malodor-forming bacteria. And in terms of R&D trends and innovation, preventing body odor has been done by either killing or inhibiting all underarm bacteria. Yet the new generation of deodorants could specifically target malodor-forming species, and companies are working on that. In terms of biofilms, we are not aware of any publication on the existence of biofilms in the axilla, although a presence of clusters has been implied in some studies. So again, at NBIC, we aim to partner this academic expertise in the area of axillary microbiome, with industry developing either a finished product, a new deodorant, or an ingredient deodorizing technology. And we have created personal care infographics, which is available on our website, and really looking at the market opportunity. So the global personal care market, we believe in 2019 was $339 billion. And we feel that a large part of that is attributable to biofilms. So we obviously create these partnering connections one-to-one, understanding well the industry needs and our capability and expertise within the academic partners. However, we are developing a collaboration portal and Big Own Innoget too. So please stay in touch for that. So a few words on NBIC and skin microbiome industries. We believe that true drivers of innovation are excellence in science and fast translation to the industry. And the goals for me are to create a consortium of leading academics in skin microbiome, to lead in academic excellence and innovative thinking in applied R&D, to better understand the unmet and also often unarticulated consumer needs, then to attract solutions that fit within the cosmetics regulatory pathways and translate the best scientific findings into industry know-how and later consumer claims. We appreciate that the understanding of the healthy skin microbiome and the complexities is in infancy, despite increasing recognition of its importance. I'll quote DSM here. So skin microbiome requires new approaches due to the structural complexity of the skin and relatively small microbial concentrations. Personal care claims are often marketing-led, so microbiome friendly, microbiome gentle, skin flora friendly, supporting the skin's microbiota and skin natural ecosystem, supporting the skin's bacterial ecosystem. These are hard to define and hard to substantiate. Yet we believe that there is a large opportunity for the personal care industry. And we also know or recognize that the quality of life which this industry supports is an invaluable asset in human existence and still economically undervalued. There are still those same fundamental questions around us. So in terms of skin microbiome, what is the healthy microbiome baseline in a specific area of the skin, be that axilla or the forehead? What are the implications of microbial diversity? So this shift from homeostasis to dysbiosis and then to full clinical manifestation of a skin disease. In terms of how the microbes communicate, do we understand enough about quorum sensing and the host response? Biofilms, well, what is the relevance of biofilm modality to skin microbiome, again, in different niches? And then what are the clinically relevant models? So how to select appropriate controls for studies? What are the appropriate sampling, sequencing, and quantification methods? And can in vitro models provide leads to clinical implications? And in these hugely complex and vast and diverse questions, we understand that perhaps the first target is skin microbial diversity and understanding the microbiome signatures in skin health and disease. So just a few examples of NBIC skin microbiome research and expertise examples. Our academics are world-leading experts in both microbiome and biofilm research. So to name a few, starting with University of Manchester, looking at skin microbiome health and epidermal lipids. University of Hull, skin microbiome health and aging. University of York and University of Edinburgh, underarm health impacting the biome. And various in vitro models for specific microenvironments, University of Surrey, Sheffield, Manchester, I'll name Glasgow there as well now. And slightly different approach, soft matter physics approach, guiding formulation design with sacrificial ingredients, reducing biofilm formation at University of Edinburgh. I would like to thank you and invite your questions.